Then let us come to the actual mechanism of enzyme action. So as we are saying that enzymes are proteins, and we know that proteins have primary structure, secondary structure, tertiary structure and quaternary structure. Here the enzymes which are proteinaceous in their nature, they are having the tertiary structure with a 3D conformation. That is, these are very much vital in the process of catalytic ability. So here this tertiary structure is useful for formation of crevices or clefts or pocket like structures, and this structure is formed because of the flexibility of the structure or crisscrossing or overlapping of that the polypeptide chains will be there. So because of that overlapping of the polypeptide chains, so we can see the formation of crevices or clefts or the pocket like structures in the 3D conformation structure of the proteins. So that is enzyme molecules, and these crevices or the cleft like structures will be acting as specific sites called as active sites. So these are the sites which the substrates are attached, or we can call them as a substrate attachment site or substrate fitting site. So in this process there are many such active sites are there. So on the enzyme molecule, so this structure of these active sites is resembling to the structure of the substrate molecules. That's the reason why that specific substrate molecule will come and bind to the active site to convert into the product molecule. So this is a way how these active sites, or we can call them as a catalytic sites also, which are responsible for the conversion of a substrate molecule into the product molecule. And here these are very much flexible and having significant or vital role in the process of chemical reactions to make the reactions to be in a short period of time and speeding up the reaction. And here, so this is a way how these active sites can get altered in the shape and structure and size depending upon the substrate molecule. So likewise there is a possibility of altering in the shape and structure of the catalytic sites or the active sites also. So likewise here the activity of the enzyme or overall activity of the enzyme depends upon the number of catalytic sites or active sites which are present on the enzyme molecule. And here these enzyme molecules, if they have more number of catalytic or active sites, they provide more space for the large number of substrate molecules so that they are converted into the products in a short period of time. And in this process, so here some changes will be taking place, where first the enzyme molecule will react with the substrate molecule. So we can show it as E plus S converted into an complex intermediate complex molecule called as a enzyme substrate complex. So this is an short-lived state and transition state, where it will not be there for longer period, but in the meantime what happens, it will be converted into one more transition state called as a enzyme product, and after this enzyme will be separated out and forming the product. Whatever the enzyme which was participating in the reaction, whatever the nature, whatever the characters related to properties of the enzymes which are there, so the same type of nature, properties and characters will be there with the enzyme which is coming out from the reaction. That means there is no change in the nature, structure, size and shape of the enzymes. So only the thing is the substrate is converted into the product molecule. Likewise here the intermediate short-lived structures are formed. So these are called as a transition structures, or we can call them as a altered structures, which are short-lived and being converted into the product molecule. So likewise these are changes which are taking place during the mechanism of the enzyme action. Here we know that different types of reactions are there, like exothermic reactions and the endothermic reactions, and here the enzyme molecule, if it have more energy, the substrate molecule, if it have more energy, than that of the product molecule, so here it is called as a exothermic reaction, where the energy will be released. And if the substrate have less energy when compared to the product molecule, so we call it as a endothermic reaction, where energy will be required or utilized. So here based on this energy relationships, so the graph is shown in which the y-axis shows the activation energy, and here the x-axis shows the rate of reaction or the progression of the reaction. Here this is a energy activation energy level which is present in the substrate molecule, and this substrate molecule may be having the more energy when compared to the product molecule, but it have to cross an energy barrier. So this energy barrier is called as a actual activation energy, and then it will be converted into the product. So the substrate and the product. So here if you show this, this shows the difference in the energy levels of substrate and the product molecule. So this is the free energy change, and this is the activation energy. So the substrate have to pass an barrier called as a activation energy barrier, then only it can be converted into the product. Maybe the product is having very less energy when compared to the substrate molecule, and if this is a condition without the substrate, without the enzyme molecule, but in presence of enzyme molecule, the activation energy level will be decreased. So here this is in presence of enzyme, so this is in absence of enzyme. So here this is the activation energy level difference between the reaction in absence of enzyme and reactions in presence of the enzyme molecule. So here this is a way how the variation can be seen, and what is happening in this process is the enzyme molecule is reducing the activation energy, so that making easy for the substrate to be converted into a product. As it is making easy for the substrate to convert into product, so the process will be completed in a short period of time. So that is the way how the reaction will be speeded up with the help of the enzyme molecule. And let us see here, this is actual energy difference between the substrate and the reaction or activation energy in presence of the enzyme molecule. So here activation energy means the energy variation that is present between the substrate molecule and the reaction process. So this is a transition state. So the substrate have to cross this barrier called as transition state. So here the average energy content, activation energy means the difference between average energy content of the substrate and the transition state is called activation energy. So like here, in absence of this enzyme molecule, the transition state will be at the higher level, so that is the energy barrier, and the transition state having decreased in presence of the enzyme molecule. That is a way how it is making easy for the substrate to be converted into a product molecule, so the reaction will be speeded up. So let us take the example here, where in the conversion of hydrogen peroxide into water and oxygen molecule. So here in this process 18,000 calories of energy is required, or the activation energy will be of 18,000 calories, but here it is in absence of the enzyme. But if the same step is taking place in presence of the enzyme like catalases, so here the energy level is decreased to 5,500 calories. So when the transition state or the activation energy having decreased, so the reaction will be taking place in a faster manner. And apart from this one, here one more example where during the catalytic activity of carbonic anhydrase, so where in the formation of carbonic acid, where it will take 200, so it will take 1 hour to produce 200 carbonic anhydrases, so carbonic acids. In absence of enzyme, it will take 1 hour to produce 200 carbonic acid molecules, but if the same enzyme is taking place in presence of enzyme, so here 6 lakhs of carbonic acids are formed in a second. So that means how much fast the reaction is going on, and here in this process we can say that it is 10 million times to that of the actual process. Likewise, the reaction will be taking fast, and it also shows that turnover number of that particular enzyme molecule. In the mechanism of enzyme action, we said that some transition state structures are formed which are short-lived and being converted into a stable product molecules. So in this process, many such changes are taking place, or altered states will be formed, altered structures will be formed. And to explain the actual mechanism of enzyme action, different scientists have tried or put forward some hypotheses or their theories. Out of them, let us discuss some of them. So let us start with the first one, lock and key theory. So this concept or this hypothesis was given by Emil Fischer. So according to this scientist, so the specificity is related with the lock and key, that is particular key will open the specific key only. So the same concept is used here, that is here the active sites on enzyme is equals to the key, and substrate is equals to the lock. So in this process, as we know that consider this as a enzyme molecule which is having the active sites on it. So these are the active sites, and this is a substrate molecule. When the substrate exactly fit into the active sites of the enzyme molecule, then it will be converted into a product which is an transition and intermediate structure. We can call it as a enzyme substrate complex. And in the meantime, the catalytic site will help the substrate to convert it into the product molecule. Where the bonds have been broken here, and it is called as enzyme product complex. Then the enzyme will be separated out as such, which have been participated in the reaction, plus we can see the formation of the product. This type of concept was given by Emil Fischer by his hypothesis called as lock and key hypothesis. So next hypothesis is induced fit theory. So this concept was given by Koshland. According to this theory, the active sites are flexible. So when they are flexible, there is a possibility of conformational change in the enzyme. There is a possibility of conformational change in the enzyme molecule. So here, when the substrate is forming and binding or bonds with that of the enzyme molecule, the active sites will undergo some sort of change. So that the change is because of the flexibility. That means the active sites will change their shape or more flexible towards the shape and structure of the substrate molecule. So likewise, if you take the example, so this is a substrate molecule and this is a enzyme molecule. Here, these are the active sites on the enzyme molecule. Here, when this substrate is ready to form bond with the active sites of the enzyme molecule, the shape of these active sites will change and they will acquire the shape of that of the substrate molecule. So that the bonding will be formed. That is what we call it as a enzyme substrate complex will be formed. So this type of conformational changes which are taken place in the enzyme because of the flexibility of the catalytic sites, this type of theory where the changes are possible is called as a induced fit theory.